Hello, everyone, and welcome to today's webinar, Molecularly Guided High-Plex Spatial Profiling in the Tumor Microenvironment. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Advanced Cell Diagnostics and NanoString Technologies. To learn more, please visit www.acdbio.com and www.nanostring.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers. Anushka Dixit, an application scientist at Advanced Cell Diagnostics, Doug Hinerfeld, a principal product application scientist at NanoString Technologies, and Chris Merritt, a senior scientist at NanoString Technologies. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Our speakers will now begin their presentations.
Thank you, Michelle. This is Doug Hinerfeld from NanoString Technologies. And just to go over the agenda for the presentation today, we're going to start by talking about the significance of spatial profiling with high plexing capabilities. I'll give an introduction to the DSP technology, and Anushka will give an introduction to the RNA-scope technology. We'll then talk about a detailed workflow for molecularly guided spatial analysis, compatibility and correlation between the two platforms, and how you can use RNA-scope to confirm results that you identify with the GeoMix digital spatial profiler. So going back a few years, Merck commenced on a study shown here. It was published here in the Journal of Clinical Investigations, and they were focused on identifying a signature that could predict therapeutic response to immune checkpoint inhibitors. And to do that, they extracted RNA from FFP samples and ran a NanoString gene expression high-plex assay. And what they identified was an 18-gene signature, which they call the GEP, which we tend to refer to as a tumor inflammation signature, as TIS. So in that graph on the bottom right, you see in the upper right-hand quadrant patients' samples that have high tumor inflammation scores on the x-axis and have very good survival, PFS, on the y-axis. The conundrum, though, is you also see patients in the bottom right where they have high levels of inflammation, but they don't respond. So the inflammation itself can be predictive, but there's still a gap in the clinical utility of that assay. They then followed this up with additional studies where, in addition to doing the tumor inflammation signature, they added tumor mutation burden as well, again, to see if they could better predict response to single-agent immune checkpoint inhibitors. So just focusing on the melanoma on the right in these results, you see on the y-axis tumor mutation burden, so patients that are high for tumor mutation burden, and then on the x-axis the TIS or GEP score. And in that upper right-hand corner in melanoma, you see that 57% of the patients that are high for both tumor mutation burden and TIS respond to the therapy. The conundrum then being is that there's still 43% of patients where these types of molecular analyses are not able to predict whether or not those patients are going to respond. So that really comes down largely to the challenge of heterogeneous tissues. So doing bulk analysis, as you see on the left, and looking at responders and non-responders, when looking at a bulk sample, you can oftentimes see very similar patterns of expression. However, if you're able to go in and target very specific regions of the tumor, perhaps immune-infiltrated regions or immune deserts, and focus just on those, you can then start to reveal interesting biology that would actually then be predictive of therapeutic response. So on the left side, there's technologies such as NanoString for very quantitative, high-plex bulk gene expression assays. And then on the right side, there's very good technologies for spatial imaging. So you get great visualization, and importantly, you retain the morphological context of the tissue. So this partnership between NanoString and ACD really seeks to integrate these two technologies. So you have the GeoMix digital spatial profiling, which is a spatial high-plex profiling technology that I'll be talking about in more detail. And then you have RNA-scope, which is a single-cell resolution gene expression technology. It's very high sensitivity and very specific. So integrating these together, where you can use RNA-scope for molecularly guided ROI selection, going to GeoMix using that RNA-scope result to identify regions of interest, and then doing high-plex profiling of those, and ultimately going back in and doing single-cell confirmation by RNA-scope-ish technology. And so I'm going to turn it over to Anushka now, who's going to tell you more about the RNA-scope technology.
Thank you, Doug. For those of you who are not familiar with RNA-scope technology, we are an in situ hybridization assay that was specifically designed to overcome many of the limitations of a traditional-ish assay, while still maintaining high sensitivity and specificity of detection. This technology consists of three major parts, as you see on the screen. The first part is our probe design, wherein ACD will specifically design a target probe against your gene of interest. The second part is your signal amplification strategy. We have a unique signal amplification strategy that ensures very high signal-to-noise ratio. And lastly, our detection and quantification strategy, which allows you to see a single molecule of RNA as a nice punctate dot. And this is available in both fluorescent as well as chromogenic platforms. The assay allows for spatial mapping of mRNAs, long non-coding RNAs, splice variants, highly homologous sequences, point mutations, and as I mentioned before, can be visualized in both fluorescent and chromogenic platforms. This assay is widely applicable to different types of samples. We can use this on FFPE tissues, on fixed frozen samples, fresh frozen samples, cultured cells, and PBMCs. As I mentioned, the RNA-scope technology has been used for the detection of a variety of different targets with spatial resolution. Some of the more specific examples include detection of highly homologous sequences like the CDR3 on T-cell receptors, GPCRs, and secreted proteins. RNA-scope has been widely applied and used for the confirmation of single-cell RNA-seq data. It's been used in combination with immunofluorescence, and the technology has been applied across vast number of research areas, which include basic science research, infectious disease research, cancer and clinical research, as well as biopharma discovery and development. Now, let's go in a little bit of more detail to understand what makes RNA-scope a unique in situ hybridization technique. The two major key aspects of this technology are the probe design and the signal amplification strategy. What's shown on the screen is our probe depicted as a Z to really tell you the different parts of this probe. Let's focus on the bottom portion of this probe, which is your target binding sequence. As the name suggests, it is a complementary sequence specifically designed against your target of interest and will bind on your target gene. The top portion is the pre-amplifier binding region, which forms the basis of the signal amplification tree development. And the middle portion is the linker region, which connects these two portions of the probe. Now, when two Z probes bind in tandem to adjacent sequences on your target transcript, it forms a Z pair. Now, this is significant because your preamplifier will come and bind onto the Z pair on your target RNA. And this preamplifier forms the foundation of your signal amplification tree. Once the preamplifier is bound, number of amplifier molecules will come and bind onto this preamplifier, which allows for the binding of fluorescently labeled or chromogenically labeled probes. Now, this will give a highly amplified signal, which allows for detection of even the really low expressing target gene. Now, this ensures the sensitivity of our technique. What about the specificity? Well, unless a Z pair is bound in tandem to a target RNA, the preamplifier cannot bind onto the Z probe. For example, if a Z binds non-specifically to another target gene, the preamplifier cannot stably bind to a single Z probe. As a result, there is no tree building and no signal detection. So this technology ensures high sensitivity and specificity of signal detection. Now, when we design a probe against your target of interest, we design it as a pooled probe of 20 Z pairs. These 20 Z pairs bind next to each other on your target of interest. But for any kind of signal to be visible, we need at least three Z pairs to bind to your target of interest. In this next slide, I'm going to elaborate a little bit about the product line that we offer. We offer RNA-scope as a part of manual assay or on automated platforms. In the manual assay, we have chromogenic assay and fluorescent assay. In chromogenic assays, we have the single-plex assay, and we also have the duplex assay available in different chromogenic dyes. In the multiplex assay, fluorescent assay, we currently have an assay that can detect up to 4 target genes on the same sample. And this can be done on FFPE tissues, fixed frozen, and fresh frozen samples. Like I mentioned, we provide assays that are supported by the Leica and Ventana automated systems. Another assay that we provide is the BaseScope assay, which is a sister assay for the RNA-scope assay, and is specifically designed to detect short targets, splice variants, and point mutations. Now, the unique thing about this assay is that it can discern up to a single base pair difference and can be very significant when you're looking at point mutations in cancer research or for circRNA detection in neurobiology. And lastly, we recently launched our high-plex fluorescence assay, which can now detect up to 12 targets on the same sample and can be very useful when samples are in limited supply. We can do this assay currently on fixed frozen and fresh frozen samples. Now, coming back to our joint workflow, we will now learn how GeoMix PSP and RNA-scope can work together to provide spatially resolved high-plex analysis. I will now hand over the platform to Doug, who will explain the GeoMix PSP technology in detail.
Thanks, Anushka. So on the left side, importantly, we have the reagents. So these are the reagents that allow us to profile large numbers of proteins or genes in spatial context on tissue. So just focusing on the protein for a minute. So what we do is we take IHC validated antibodies that are specific to the targets that we're interested in analyzing in the tissue, and we conjugate to that a UV photocleavable linker. So every antibody gets a unique DNA oligonucleotide with this photocleavable linker conjugated to it. The same is true for the RNA probes. So we take ISH probes. To each probe, we conjugate a unique photocleavable linker. What that allows us to do is, again, back to the protein, take a bunch of different validated antibodies, each with a unique linker, pool them together, stain the sample, and then it's ready to go onto the GeoMix platform. The GeoMix platform essentially is a microscope, and it has the unique ability to shine UV light exactly where you want it within the tissue, and only where you shine the light are those tags released. Those tags are then collected and analyzed in a spatially distinct way, as shown in the data on the right, where you're looking at the expression of the different antibodies, the different markers in the panel across these different distinct locations within the tissue. So preparing a sample for GeoMix is very straightforward. For protein, it's essentially the same as immunohistochemistry, and for RNA analysis, the same as in situ hybridization. So we combine two different reagents. What this is showing here on the left is the imaging reagent. So these are RNA-scope probes targeted to genes of interest. And then you combine those with the profiling reagents, which are the DNA barcoded-ish probes that are used for the nCounter or GeoMix readout. Once you've stained the sample, it is ready to go on to the system. So on the left is the GeoMix platform itself. It is a 4-channel fluorescent microscope. So the slides are inserted into the system. It then images it for whatever channels you're using. You then use that image to define regions of interest throughout the tissue. Once that's done, the system shines the UV light only on those regions of interest, one at a time. So it shines the light, zips up the tags, takes the tags from that specific region of interest, deposits it into a plate, goes to the next region of interest, shines the light, zips up the tags, puts it in the plate. So it goes around and around very quickly and cycles through the image and profiles each region of interest. That plate then comes off the instrument and gets hybridized to our nCounter barcodes, which I'll talk about in a minute, where each of those UV cleavable barcodes are actually counted and enumerated. So how do we count these molecules? So this uses really the bread and butter NanoString barcoding chemistry. On the left, we have a capture probe, and on the right, we have a reporter probe. In the middle are these half sites. Each of these half sites are complementary to a very specific sequence. So if that sequence is present in the sample, when we hybridize it together, it forms this complex, and then that complex is read out on the nCounter platform. So you'll see in this case, this sequence that you're hybridizing the capture probe and the barcode to is in fact this photocleaved linker that's in the well of that plate from the system. Once we've done the hybridization, we run it on the nCounter. The nCounter is also a microscope, and it images and takes pictures of these barcodes and simply counts them. So every time a particular protein was present in the sample, its barcode was present. We're reading the barcode, and then we know that that protein was expressed at that particular location. So a really simple example of that to give you a feel for how it works. So here we have lymphoid tissue. It's stained with a collection of DNA barcoded antibodies, and at the same time stained with both Ki67 and CD3 fluorescent antibodies. We image it on the system. We then select regions of interest. So in ROI 5, you see here, that is an area that is fluorescently very strong for Ki67. And the DNA barcoded antibody for Ki67 then shows up by reading the digital counts shown on the y-axis. The same is true in ROI 9, but for CD3. But remember, we're staining with many more DNA barcoded antibodies in this case, which you don't see at this level. But once we count those, you then see and are able to quantitate the expression of all the different markers that are in the panel that you're using. So you can see on the y-axis the different antibodies that were analyzed and then the different regions of interest, and you can see how they cluster together into very distinct and heterogeneous profiles. So another really unique aspect of this technology is the ability to exquisitely control where on the tissue you shine that UV light. And the way we do that is we've incorporated a digital micromirror device. So each one of these little independently controlled mirrors is about a micron square in size. And what that allows you to do is to shine the UV light onto that digital micromirror device, and it shapes it onto the tissue. So in this example, we've stained with pancytokeratin and then DNA in blue. And we create a mask, so all regions that are positive for pancytokeratin, and we shine the light there. We then turn around and shine the light just on the pancytokeratin negative region. So essentially what we've done is optical microdissection of the tissue in this sort of organic phenotype-driven way. So going beyond that one approach, this allows you a lot of flexibility in how you interrogate the sample with spatial resolution. So you can do simple geometric ROIs, looking at regions of interest, perhaps infiltrated tumor or immune deserts. And this is a method that's been very successful. We've had a number of very high-profile papers already published using that approach. You can also do the segmentation, like I just described, focusing just on pancytokeratin positive or pancytokeratin negative as an example. The rare cell application is an excellent application when you really know a specific cell type you're interested in profiling. So, for example, you might be staining for CD68, and then the dark cells here would be the CD68 positive cells. You would shine the light and profile only those CD68 positive cells, and then you can profile the CD68 negative cells or the white cells. It does all of those cells at the same time, so that digital micromirror device allows you to shine the light only on those dark cells and nowhere else, and then only on the white cells and nowhere else. Contour profiling is another application that's been interesting both in cancer, but also there's been a lot of interest in this in the context of neurobiology. And what this allows you to do is essentially profile the tissue different distances from some biological structure or cellular structure of interest. So it could be a invasive margin or something like that. And then the gridded profiling is an application where, you really want to just uncover the heterogeneity of the expression of all the markers in the panel across the tissue. So it's a much less targeted approach, but a great way to discover the heterogeneity of expression of, say, the immune landscape within the tissue. All of this is really integrated into an excellent, very easy to use integrated software solution. So on the left, you have the control center. That's what allows you to do the imaging or scanning of the tissue, select your ROIs, initiate your collections. Once that's done and you have that plate that's hybridized to the NanoString barcodes, that then goes out to the in-counter for counting. That data then comes right back in to the GeoMix data analysis software shown in the upper right, where you then retain the association of the images, the regions of interest, the probes, and the final data and data visualization, all in the same software. And it's built in a cloud architecture. So while it runs on the system, anybody from their computer can essentially go to Google Chrome, log into the system, and can do many different aspects of the data analysis and ROI selection there. So what this shows on the left is you have a technician at the lab who's running the instrument. They've loaded the samples, they've set up the scans. They then maybe notify the pathologist. It could be in another building, could be in another site within your organization. They then select the regions of interest and do the segmentation. Once that's done, they can approve the ROI collection. It then goes about collecting the ROIs. Once the technician has brought the count data back in from the in-counter, people can, the principal scientist or the biostatistician, they can all log in, review the data. They can all be reviewing that data while someone else is collecting another study. So it's a great platform for collaborative research where, you have a technician, you have a pathologist, you have an oncologist, you have a biostatistician. They can all collaborate really well to work within the platform. In terms of the content, so we've put a great deal of effort into building out really useful, validated content. And just to give you an example, on the upper left, on the protein side, what we've done is we've built an immune cell profiling core. So this is a 20-marker core that has all the positive and negative controls, and then it has all the things like CD3 and CD68 and CD45 and sort of the regular players there in the immune system. And then you can select different modules that you want to run with that. So we have a pan-tumor module, an immune cell typing module, an immune activation status, an IO drug target, and we continue to expand these modules. But importantly, you can add custom content. So if you have an IHC validated antibody, it could be an antibody that's very proprietary to your research, your drug target, that's fine. We can put a unique DNA barcode on it, validate it, and then spike it into the panel. And so you can add custom content as well. On the right is the RNA assays, the spatial RNA, which is really a large focus of today's discussion. So we really just dipped our toe in the water here. We have an 84-gene immune pathways core, and that is focused on human IO and looks at things like immune response. as well as that 18-gene tumor inflammation signature that I discussed at the very beginning in the Merck paper. And then you can add custom content to that as well. So one of the expansions and flexibilities of the platform is that, if you recall, you collected those DNA barcodes into a plate. You can hybridize those to the nCounter barcodes and analyze them on the nCounter. But you can also take those and make sequencing libraries. And what that does is it creates more flexibility and enables more discovery type applications. So you're able to increase the numbers of analytes that you want to look at. In this case, we are designing an initial panel of about 1,500 genes. And the content is drawn from our very popular pan-cancer pathway panels that are shown here. So again, the platform allows you, and in this case, we're looking at protein data again, to really understand the heterogeneity of the tissue. So in the top left, this is lymphoid tissue. We've selected different regions of interest based on the staining of CD3, CD20, a B-cell marker, and pan-cytokeratin. And you can see, looking across the x-axis, the different analytes, looking across the y-axis, the different regions of interest. And you can see the extensive heterogeneity of expression across the tissue. And how similar regions cluster together. The same is true on the right in colorectal cancer. So we have RNA profiling, we have protein profiling. We can actually do both of them. In that case, what we're doing is we're using serial sections. So we're doing 40 proteins on one section and then looking at the same exact regions of interest. So registered regions of interest, we're looking at the expression of 84 RNAs. And this is a small study in colorectal cancer. Here we're staying with pan-cytokeratin, CD45, and CD3 to guide where in the tissue we want to select these regions of interest, as well as the architecture, looking at the invasive margin and so forth. And then on those serial sections with ROIs placed essentially in the same location, we segment it into tumor and stroma, so you can see that in pink and green. And then we can look at the expression of both the RNA and the protein. And so these are shown in two different heat maps here, where if you look at the top bar there in the green and in the pink, you can see how the tumor segments cluster largely together and the stromal segments cluster together, kind of turn 90 degrees. You see the same with the protein. Interestingly, you do see some of the tumor segments that cluster more with the stroma. And when we went back and looked at those were actually highly infiltrated tumor regions. So they had a lot of those sort of stromal and immune markers within them, which caused them to cluster to look almost more like stroma. So with that NGS readout capability, we can really start to look at hundreds and thousands of genes spatially expressed. And what this allows you to do is to look, for example, if you look at the bottom two panels here, we're doing spatial analysis, and we're looking at two regions of the invasive margin and looking at these pathways and the activity of these pathways, you can see very different responses. So on the right, you have sort of a very inactive, quiescent invasive margin, and then on the left, you have a much more active or invasive margin. So you can really start to understand the biology, understand how these immune pathways are activated, which pathways are activated with spatial context. So I'm going to turn this over to Chris now, and he's going to talk about expanding the GeoMix DSP capabilities and applications with integrating the RNA-scope technology.
All right, thank you, Doug. This is Chris Merritt. I am in R&D at NanoString Technology. So our goal was working to expand the capabilities, as Doug said, of GeoMix and RNA-scope, and basically combining these two strong technologies together for one workflow. So this is just a cartoon, a little image of the workflow that we worked out. So in the end, on the upper left-hand corner, you can see the probes that we use for RNA-scope that Anushka went through, those double Z probes. We found that if we hybridize those first in the workflow in the like auto stainer, then move on to GeoMix DSP probes, we got the best results. So the workflow is laid out here where we do RNA-scope the first day, GeoMix DSP probe hybridization overnight into day 2. In the middle, we do GeoMix DSP on through day 2, and then we do the quantification on nCounter on day 3. But this is a really nice automated workflow we had worked out. So I'm going to show some data showing how these two platforms play well together. So on the left here, what we wanted to look at first was the RNAscope signal remaining the same when we add GeoMix RNA assays on top. So these are control cell pellets I've shown here with 3 housekeeper controls. So in the upper section, so I'm looking at the left-hand side where it says RNAscope only, you can see bright signal, green, yellow, and magenta for the 3 targets. Then to the right where we do RNAscope plus DSP RNA, you can see similar signal intensities. Below that, there are the DAT-B negative controls. Again, with RNAscope only, you see no signal, and not surprisingly, when you add DSP RNA on top of it, you no longer have signal. So at a high level, the signal seems unchanged, which is great. And so to the right, we're basically doing the inverse. Is GeoMix RNA signal altered when we add RNAscope on top? So again, this is just a really large field of view of a mixed cell population. And basically the graph on the right shows if we add RNAscope on top of GeoMix DSP and compare it to GeoMix DSP without RNAscope, we get indistinguishable results. So that was nice. So next thing we wanted to do is basically bring it out of our model systems and try this on tissues. So first I'm going to show some tonsil tissue, and on the left here is an image of a tonsil with 12 ROIs that we had profiled. So these are they're small circles that you can see on the left, and then these are blown up in the middle panel. So we used RNAscope visualization reagents, CD3E, which is in green, and CD19 in magenta, and we also put a DNA dye on there, which is in gray. So we use these RNAscope markers to actually help guide our ROI selection. So if you can see in the middle here, you kind of see a blend of CD3E and CD19 populations. And then on the right, what I'm showing are some correlation graphs of the DSP results and the ACD results, or the RNAscope results. So in the upper right-hand corner, where you have the magenta dots, I'm comparing 2 B-cell markers, which we'd expect to correlate. So CD19 is on the x-axis. So CD19 RNAscope mean intensity. So I just pulled that right off the DSP instrument to get the mean intensity. And then on the y-axis, we have CD20 GeoMix DSP signal-to-noise ratios. And what's really nice here is that when you compare the 2 data sets from the same tissue section, you have a really high correlation, high R-squared value. Below that, in the green graph, is CD3. So this is actually the same target that we're going after. So this is CD3E. Again, bottom axis, RNAscope mean intensities versus GeoMix DSP SNR have a high correlation of R-squared is 0.98. So that was great in this tissue. The second tissue we looked at was non-small cell tissue. So again, looking at the exact same targets, CD19 and CD3E. On the left, you can kind of, basically it's a high-level view of this non-small cell lung cancer tissue. We went in and profiled 12 ROIs again, which are these circles, blown up in the middle. And then, again, we were basically, for this proof of concept experiment, looking for a mix of B cells and T cells. So going on the far right, and the upper panel, the upper graph with the correlation, again, looked at CD19 and CD20. Again, really strong correlation between the 2 platforms. And this is, data on the same section. So an R-squared of 0.98. And then if you go down below, we have the CD3 correlation, which in general is a little less than CD19 and CD20, but it's probably mostly driven by the lack or the very low proportion of T cells in the sample. But again, what was really nice, we can run both assays on the same section and get really nice data that actually correlates quite well. So moving out of those model systems, so now we have confidence that these 2 assays work well together. We wanted to actually move this into IO research and something of more interest to the general community. So generation of immunity is a cyclical process that's shown on the left here. And what we're looking at is we want to basically characterize the cytokine expression and the tumor microenvironment in these samples using both of these technologies. So on the right, we have a little workflow. So we're going to use ROI selection for RNAscope, which I basically showed in the last two slides, and then we'll do DSP analysis on top of it, again, as I showed previously. And then Anusha is going to come in and actually show some confirmation of GeoMix DSP results using RNAscope. So basically going full circle with these two technologies. So this is the workflow again. To reiterate it, low-plex RNAscope determine the regions of interest. In the middle, we're going to do GeoMix DSP and then confirmation. So the first bit here is the low-plex determination of regions of interest. So here's another non-small cell lung cancer tissue we're looking at. So this is different than the one I showed previously. So in this sample, we again profiled with CD19, our B cell marker, and CD3E, which is our T cell marker, and DNA, just to guide selection. So in this case, we actually profiled 24 ROIs, and then on the right there, I again zoomed in on these to show the different kind of populations there. In the upper left, around ROI 1 through 6, you see really strong expression of CD19, so those are really rich for B cells. As we go down more towards like that bottom row, we enrich more for T cells, or basically are going after tumor regions that have very little immune cell content. So again, these images were beautiful and are really helpful to actually guide the selection on GeoMix DSP. So what we did next is we ran that experiment that I just showed there on the encounter system. And as Doug mentioned previously, you get a lot of data from this on the encounter system. So this is 84 genes from the IO panel, and the names are too small, but I'll blow up some of these. On the far right, they're gene names for all 84 genes. On the left there, again, I'm just keeping the image there for reference. So we looked at 24 ROIs, and those are all listed along basically the axis of this heat map, of this cluster heat map. So I highlighted a few different regions, so CD19 enriched, immune poor, and CD3 enriched ROIs. And basically what I am showing here is these are Z-scored across genes, and these are basically clustered in an unbiased way. And you can see the immune poor, CD3 enriched, and some CD19 enriched ROIs do cluster together. And then highlighting some of the genes that we're actually seeing in these ROIs. So for the CD19 enriched ROIs, we're getting the characters we'd expect. So the HLAs, CD40, MS4A1, which is CD20, and then CD74 and other targets. And then on the far right, I'm highlighting some of the CD3 enriched ROI expressors. So CD4, CD8 expected, CTLA4, TIGIT, CD3, FOXP3, VSIR, which is VISTA, and LAG3. So that gives us a lot of confidence that we're actually profiling these appropriately with DSP. And then I also finally highlighted in the box right in the middle, in the immune poor ROIs, is that large blue square of just low expression. And that, again, these are ROIs where basically there are very few immune cells that we could see based on our visualization with RNAscope. And again, blue is basically very low expression, as we'd expect there. And now, so for the last step, we actually went in and confirmed some of the results with RNAscope. So I'll hand it off to Anushka to go over that.
Thank you, Chris. As Chris very nicely explained, RNAscope was used to initially identify the CD3-rich versus the CD19-rich regions in the lung cancer tissue, followed by which DSP analysis provided a very extensive gene expression profile for 78 targets in multiple ROIs. As Chris said, this is a lot of data, and in this last step, what we do is, since RNAscope can provide truly single-cell resolution, we can use RNAscope to confirm expression of a handful of target genes that you might be interested in. So we can really hone in on specific regions of interest and confirm the expression of genes that might have functional significance while providing spatial orientation in your tumor tissue. So out of the 24 ROIs that were assessed by DSP GeoMix, we compared the gene expression profiles between two ROIs, ROI11, and ROI6, and validated the expression of a few genes that are significant in immuno-oncology research. As you can see on the screen, ROI6 is a CD19-rich ROI, whereas ROI11 predominantly is a CD3-rich ROI. Now, the genes of interest that we selected for confirmation are significant in the cancer immunity cycle. I'm sure all of you are familiar with this cycle. With the rise in success of immunotherapies in recent years, there's a need more than ever to assess the different cell types involved in regulating this cancer immunity cycle. These include cells that produce soluble factors like cytokines and chemokines, which promote and activate T cells. They help in T cell proliferation and also migration to inflamed immune sites. So some of the targets that we picked include CCL5, which is a chemokine secreted by NK cells, CXCL9, which again is a chemokine that attracts dendritic cells and other immune cells to the inflamed site. Interferon gamma, a cytokine secreted by activated T cells and other immune cells, and CD8, which is a marker of the effector T cell. So with RNA-scope analysis on these 4 targets in a serial section of lung tissue, we observed that all these 4 genes were expressed at a very high level in the ROI11. And this is exactly what we expected, since ROI11 is the CD3-rich region. But having said that, we do see some basal level of expression in the CD19-rich ROI, ROI6. Now, let's focus on the right-hand side top panel, which shows you a magnified image of the ROI11. And here, you can really appreciate the single-cell resolution provided by RNA-scope. The cell marked with the blue arrow is a T cell that secretes CXCL9, CCL5, and is positive for CD8. So this is potentially an active T cell that's trying to secrete cytokines and activate other T cells in its vicinity. One of the most important things I think to take from this slide is the correlation between the DSP data that is shown in the graph in the bottom panel and the staining of RNA-scope for these 4 targets. And this really tells you that both these techniques have concordance and can synergistically be used in a workflow to spatially map gene expression. In addition to analyzing some of the activation genes, we wanted to look at some of the immunoregulatory targets, which are common targets for currently popular immunotherapies. Now, these targets include CTLA4, PD1, which are inhibitory receptors on T cells, and PDL1, which acts as a ligand for this receptor. We also looked at ICOS-LG, which is again a ligand for a co-stimulatory receptor. Again, in this image, you see all these 4 target genes probed together on a serial section. You can see the staining very dramatically positive in the CD3-rich ROI11, but you also see moderate staining in the CD19-rich ROI, which tells you that these cell types are definitely present in both the ROIs, but at a much higher level in the CD3-rich ROI. Again, the concordance between the DSP data and the RNA-scope staining data is pretty good, which is seen on the graph in the bottom panel. And in the top right panel, again, a magnified image of the ROI11 really highlights the PD1 positive, CTLA4 positive T cell that you can see. So comparing the gene expression profiles of some functionally relevant genes within different regions of heterogeneous tissue, we can identify potential biomarkers that can really help stratify patients and perhaps predict immunotherapy response. So what have we learned today? With this joint workflow, what we have shown you is that we can spatially map gene expression using RNA-scope and identify focused regions of interest in a tissue. Next, we can obtain a gene expression profile for focused areas using GeoMix DXP. And finally, for a handful of regions or for a handful of genes of interest that you want to hone in on, we can visualize specific targets in the tissue with morphological context and single-cell resolution using RNA-scope. So with that, we know that GeoMix PXP and RNA-scope are individually very powerful techniques, but with the joint workflow, we can have spatial gene expression profile with high plexing capability while retaining the morphological context, which is extremely significant for heterogeneous tissues like the tumor, especially lung cancer tumor. And with this joint workflow, we have combined the best capabilities of both these technologies while maintaining technical feasibility for each of this assay. So with that, I want to thank all of you for your attention, and we'll take any questions at this moment.
Thank you, Anushka, Doug, and Chris, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Our first question is, what is the spatial resolution of the GeoMix sequencing?
Yeah, thanks, Michelle. This is Doug. I'll take that one. So the platform using the DMD, the mirrors, allows you to really shine the light down to very small features within the tissue. You can certainly shine the light down to an individual cell. It really depends on the application of the platform and the research interest that you have and the sensitivity of the particular markers you're interested in. So it's more common that people look at very specific cell populations within distinct spatial regions of the tissue. Thanks.
Thank you. All right. Your next question, can high-plex RNA-scope be used as GeoMix morphology markers?
Hi, thank you for the question. Right now, the high-plex is available for fixed and fresh frozen samples, and so we recommend using our multiplex probes for ROI detection with GeoMix right now.
Thank you. Okay, your next question. Can we use more than 2 genes for the ROI selection?
Thanks for the question. Yes, we can do up to 3 genes for ROI selection as the first step in this workflow.
Thank you. Okay, well, we're getting so many great questions. Okay, next is, let's see, how many targets can we analyze at a time using the GeoMix DSP?
Yeah, so as I was talking about earlier with the capability of the next-gen sequencing readout, we haven't actually found the limit. We've certainly looked at over 5,000 distinct ISH probes and been able to very accurately quantitate that large number of genes. So we have not found the roof, and you know, with sequencing capacity, it's certainly quite high.
Thank you. On an average, how many BP long is each Z probe?
Hi. So a single Z probe can bind to around 18 to 25 base pairs on the target. So on an average, I would say 25 for each Z probe. So 50 for a pair.
Thank you. Okay, your next question. What kind of method do you use to validate your RNA-scope data?
Hi, can you repeat the question?
Sure. What kind of method do you use to validate your RNA-scope data?
So if you want to assess the specificity of the staining, you can use a positive and negative control, which are specific probes that we provide with the kits, and that way you can really discern between specific and non-specific staining.
Thank you. Okay, let's see. Next question. Let's see, we're getting so many great questions. Okay, is RNAscope a tissue non-destructive technology?
Yes, it does not destruct the tissue when you probe with RNAscope. It is non-destructive, so your tissues remain intact at the end of the assay.
Thank you. Okay, next question. How many tissue slides does the GeoMix system hold at one time?
It holds 4 slides at a time. So you load 4 slides, scan them, select ROIs, collect, and then you load the next set of slides. And you can do multiple sets of slides in a day.
Thank you. Next question. Could you identify the RNAscope intratumoral clonal population of T cells?
Yes, you can actually use our BaseScope assay, which, like I mentioned, is specifically designed to identify highly homologous sequences. And that way you can have a specific CDR or T cell receptor probe that we can design and discern different clonal populations using our BaseScope assay.
Great, thank you. Okay, next question. Can RNAscope be combined with immunofluorescence for the validation step?
Yes, we can combine RNAscope staining with immunofluorescence, and we have this joint workflow tech note available on our website as well. This is a method that's very popular, and a lot of our customers use it often for their research.
Thank you. How is SNR from the GeoMix different than the normalized counts generated from typical N-counter output?
Yeah, I can take that question. So the SNR is different from the typical N-counter output. So just based on time constraints, we didn't go through all the details of the RNA assay. But within the 84 Plex gene IO panel, we actually put in 6 negative control probes along with the 84. So those are basically like alien or scrambled sequences just to measure the background that we get for a typical probe. So we use those just for in situ. The background's very different than a normal in-solution hybridization like N-counter. So we use that as the background measurement. And the SNR is basically the counts we're getting from, say, CD3 divided by the average of those negative control counts.
Great, thank you. All right, next question. Can DSP RNAscope be used to detect mRNA or DNA with a hotspot gene mutation in a specific cell type in a tissue section?
So maybe I'll go ahead.
Sorry, go ahead, Doug.
Okay, I was just going to say that as long as you can, so if you can fluoresce the target of interest using, in this case, RNAscope, then we can use that to guide the cells in which we analyze with DSP. So, Anushka, maybe the question is partly about detecting point mutations fluorescently?
Yeah, so I was going to say that with RNAscope, we cannot really detect DNA, but we can detect mRNA targets, and for specifically point mutations, we encourage using BaseScope, which is chromogenic and not fluorescent. So you can validate your target expression, but not use it for ROI selection.
Thank you. I'd like to once again thank Anushka, Doug, and Chris for their presentations. Do you have any final comments for our audience?
As Michelle mentioned, feel free to reach out to any of the our emails on the top of the page there that you're looking at now, and we'd be happy to answer any additional questions that you have.
We also have a link to our joint white paper that we published a couple of months back, and that is available on both of our websites, and the links are provided on the slide.
I'd like to once again thank our speakers for their time today and their important research. We would also like to thank LabRoots and our co-sponsors, Advanced Cell Diagnostics and NanoString Technologies, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. LabRoots will alert you via email when this webinar is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you.